In this video, we'll explore what live blood analysis is and examine the impact the atom cell device had during this type of analysis. Live blood analysis is a screening method that allows us to observe the various components of a fresh blood sample under a microscope. It provides insight into the functionality of the blood system and can reflect aspects of a person's overall health. Human blood is made up of approximately 45% cellular components, namely red blood cells, white blood cells, and platelets, and 55% plasma. Blood performs many vital functions, including transporting oxygen, regulating body temperature, and supporting the immune system. The condition of the blood often correlates directly with our overall health and can be influenced by diet, lifestyle, and environmental factors. During a live blood analysis, a small drop of blood is obtained using a finger prick. The sample is placed directly onto a glass slide and viewed immediately under a microscope at up to 1,000 times magnification. The lighting technique used allows even the smallest details of the blood to be seen clearly. This method enables practitioners to observe the structure and behaviour of the blood components in a near-living state, similar to how they function inside the body. In this study, an initial blood sample was taken from the test subject. The subject's food and fluid intake had been monitored, and no earpods had been used prior to testing. The first sample revealed a healthy blood profile. Red blood cells were freely flowing, with healthy cell membranes and clean plasma. 30 minutes later, the subject wore a pair of active earpods for 20 minutes. 10 minutes after exposure, a second blood sample was taken. This sample showed a compromised blood profile, with pronounced rouleau formation, the stacking of red blood cells, and significantly inhibited blood flow. After another 30-minute break, the subject wore the same active earpods for an additional 20 minutes. However, this time, the atom cell was worn on the body during exposure. 10 minutes later, a third blood sample was taken. The results were notable. The third sample showed a marked improvement in the blood profile, surpassing even the initial baseline. Rouleau formation was no longer present, and healthy blood flow had been restored. The images from the analysis suggest that wearing the atom cell may play a protective role against the adverse effects of electromagnetic radiation on blood health. The full dataset from this study is available for viewing on the research page at atomlab.uk.